Hello, welcome to High Five Oncology. On behalf also of Nadia Harbeck and myself, would like to welcome you to this take from the San Antonio Breast Cancer Symposium 2024 with a distinguished faculty, Peter Fasching, Barbara Pistilli from Paris, Janni Luftner from Berlin, and Michael Untch also from Berlin. We're going to discuss two abstracts on the HER2 positive breast cancer entity, and I would like to start with a neoadjuvant trial, which was introduced by a Chinese working group, looking at a Chinese ADC as well as a Chinese TKI. It was a prospectively randomized study looking at three arms, standard of care, TCHP, which standard of care probably globally, versus the Chinese ADC, versus the Chinese ADC combined with peritoneum. In my view, it was a pretty fascinating study. Actually, the study was called FASCINATE. Did it also fascinate you, Barbara?
Yes, I think that the study deserves this name. First, because it's a new antibody-drug conjugate. It's a new antibody-drug conjugate that has a target HER2. So the antibody backbone is trastuzumab. The linker is a cleavable linker. The payload is always a topoisomerase 1 inhibitor, but a little bit different as compared to some of the topoisomerase 1 inhibitor that we are in some of the approved ADC. It looks like it's more permeable, so it can provide a greater bystander effect. And this study is very interesting because it's one of the first study that is releasing the results of the use of ADC in the neoadjuvant setting, and particularly in patients with HER2-positive breast cancer. The results are encouraging, I would say promising, because the PCR rate is absolutely comparable to the PCR rate obtained in the standard arm. I would make just a couple of comments. The first one is the fact that the patients that received the SHRA1811, that is the name of the ADC, received 8 cycles of this treatment, either as a single agent or in combination with peritoneum, whereas the patients that received chemotherapy, standard chemotherapy, received only 6 cycles of chemotherapy. So I would like to understand better why the duration of the treatment with ADC was longer. And the second comment that I would make is that the toxicity profile of the combination with peritoneum was quite high, with 70% of grade-free adverse event. So for sure, this is a kind of combination that we cannot consider for further study or for a treatment that we can use in clinical practice. But the use of this ADC as a single agent, I think that it deserves further investigation and to be tested eventually in phase 3 trials.
Well, thank you very much for these impressions. Just a very quick question. Were you surprised that the combination of peritoneum and the ADC was no better than the ADC alone?
Yes, I was surprised, but at the same time, I think that I'm not sure about the rationale of this combination. We know that the combination of some TKI, HER2 targeting TKI, potentially can increase the internalization of the ADC, but I'm not sure that behind this specific combination there is a strong rationale.
Well, thank you very much. Michael, there was quite a bit of Chinese presence here. Excellent presentations, some very, very interesting data. Is this the beginning of the Chinese era?
For sure, yes. And Barbara, I will just take one of your comments as a thought on what is happening. The combination of an anti-HER2 antibody with a receptor tyrosine kinase inhibitor is not so young, because we did it in the neo-ALTO study, we did it in the ALTO study, we all know the results. We also used an extension of an receptor tyrosine kinase inhibitor with neratinib. So I think the idea which was proposed by our Chinese colleagues was quite a good one. And as you have shown, the antibody-drug conjugate did it alone, I would say. We use for the first time an antibody-drug conjugate in HER2-positive breast cancer patients in the Catherine study, in which last year we presented for the first time the overall survival data. We are having or are waiting for the results of TDXD versus TDM1 in DESTINY-Breast04 post-neoadjuvant in patients with residual disease after neoadjuvant chemo and anti-HER2 therapy. Nadia, you know that we are expecting also the data from DESTINY-11. So you maybe make one comment to that. So the Chinese are coming, and I have been in ESMO on a Sunday afternoon in the Gyn Onco session, and it was from 7 fantastic presentations, 6 were coming with ADCs from China. So I'm fascinated by the development of our colleagues over there. And we are already cooperating with them. So I'm using it right now in 3 studies, antibody-drug conjugates from Chinese developers. And I think we should follow the track here and keep this cooperation ongoing. So again, very nice data, nothing more, because we have to wait for other concepts and greater or powered studies. I would say, yes, the Chinese are here, and we should cooperate with them.
Thank you very much. You might consider starting learning Chinese, Michael. But now let's talk about another study in metastatic breast cancer, that is in hormone receptor positive, HER2 positive metastatic breast cancer, that we saw data from the PATINA study. PATINA study randomized patients after induction chemotherapy in combination with HER2-targeted treatment versus endocrine maintenance therapy in combination with HER2-targeted treatment versus endocrine combination treatment, including palbociclib, together with HER2-targeted treatment, of course. And it was a positive study with a PFS benefit in favoring the endocrine combination, the addition of palbociclib. No overall survival benefit right now so far. What's your take on these data, Peter? And especially, do you think that we have to offer our patients now in future after standard chemotherapy or standard therapy, according to Cleopatra, the endocrine combination maintenance?
Yeah, I thought you picked that study for discussion. We had some data, including your own DETECT-5 study in that setting already, but I think the PATINA study pretty much closed that chapter looking for an efficacy of CDK4/6 inhibitor and HER2 positives. And it really warms my heart because if you look back in 2009 in the original publication, for the preclinical data for palbociclib and the efficacy is already known for a very long time that there's a very good efficacy of CDK4/6 inhibitors in patients with HER2-positive disease. And having a chemo-free option that is very efficacious, and the median PFS time is getting over 40 months. That's very impressive. Of course, is something that you want to have for your patients. So if you say, could this be a standard? Who would not want to have a chemo-free regime where you get over 40 months? 2 more comments to that. I think, though, that it's a very different approach in HER2 positives, and maybe the lacking overall survival benefit or the magnitude of benefit already points at it. Usually in the HER2 positive studies, when we improve the anti-HER2 treatment, we see a very, very good correlation between the PFS and the OS. So most of the PFS benefit translates into an OS benefit. We don't see that here in the PATINA study, and I wouldn't expect it because with the CDK4/6, it's not an anti-HER2 treatment. It's the blockade of an additional pathway. And I find it quite interesting because it opens up a kind of complete new chapter in anti-HER2 treatment because we haven't done that yet so far. We've either done anti-HER2 treatment, we've done chemotherapy, but now something else is added on top of that that we might have to work on a little bit in the future to better understand.
Well, thank you very much. I'm glad that it warmed your heart. This is very good before Christmas. Diana, what's your comment on this study? Will it change your practice?
I think it's necessary at least to talk to the insurances and to try to take over the costs, at least try it. But for the future, I would say I have 2 ideas in my mind. First of all, you mentioned it in your initial sentence is that the randomization was done after chemotherapy. My first question is, can we skip chemotherapy? Of course, this is a selected patient population where primary progressive patients were taken out. But is the efficacy high enough that you would really skip the introduction with the chemotherapy? That's the first thing. The second thing, we all know that TDXD has a very, very high probability to enter the field of first line. On the other hand, the toxicity may lead us to some kind of de-escalation, like a dose reduction, or even change treatment to an endocrine-based anti-HER2 therapy-based maintenance. This is the two things that I am guessing about because we do not have any data, but it's very seductive to introduce this into clinical practice. But I'm talking to somebody who is a little step further, because you have done the DETECT 5, and my idea would be that you are pretty much in favor of, skipping first-line chemotherapy.
We'll have to see. We showed the data, as you know, at ESMO, and they were encouraging that we might, at least in some patients with triple-positive metastatic breast cancer, skip chemotherapy, especially if we add the CDK4/6 inhibitor. Well, Nadia, what's your closing remarks?
I think we heard one study, Patina, which is going to be practice-changing. I think the data were so fascinating, and Sarah Hurwitz said that in her commentary that she was overly enthusiastic, I think, but she brought it over quite nicely. So, but Michael said it before, next year will change again, because we will probably hear DB09, DB05, and DB11. Then we can sort of rewrite the story about HER2-positive breast cancer, be it with or without endocrine-based backbone. So I think a lot of interesting data coming. For the time being, I think Patina will give us something to talk to our patients and probably also to the insurance companies about. And actually, we've done this already in some patients where they had heterogeneous tumor biology, like coming from the early breast cancer setting with luminal-like behavior, and then adding HER2 in the metastatic setting. And for those patients, we've already done this concept and didn't even ask the insurance company, and they had wonderful results. So I think something to look forward to. And I don't see any further comments. So with that, thank you very much for discussing HER2-positive breast cancer, and thank you all for watching, and stay tuned. We'll have other subtypes